Hello students, how are you? Welcome back to Nucleus Neet coaching. I am Kiran Kumar, your biology NEET faculty. Students, today we are going to continue the remaining syllabus from the chapter Cell, the Unit of Life. So in this session, I am going to demonstrate how chromatin, which is present inside the nucleus, can form a chromosome by using a rope. Okay, so here I have some rope with me. This rope you have to assume as chromatin, which is present inside the nucleus. Okay, this chromatin later it gets folded up and it forms a chromosome. How this actually happens inside the nucleus, I am going to demonstrate you in this video session. So watch this video up to the end without skipping. Okay, students, you know how the toppers get seats in the topmost medical colleges like AIIMS and JIPMER. They will understand the concepts crystal clear. So in this session, I am going to make all the concepts crystal clear. So it is very valuable. You will not get this content anywhere in the YouTube. So take pen and book, get ready for the class. Students, what is nucleus? A nucleus is a very important cell organelle which is present in all the eukaryotic cells like plant cell, animal cell, fungal cell and protist cell. In which cell you don't find nucleus? Exactly, nucleus is not observed in prokaryotic cells. Okay, so inside the eukaryotic cell, the nucleus, it controls all the metabolic activities of the cell, all the living activities of the cell. That's why a nucleus is called the brain of the cell. The brain of the cell. Just like our brain, which controls all our organs in our body, the nucleus present inside the cell, it will control all the metabolic activities, living activities of the cell. So that's why there is one more name that is called director of the cell. It is also called director of the cell. Director of the cell. This is another name for nucleus. Okay. So the nucleus was first described by a scientist called Robert Brown. Robert Brown. Don't get confused with another scientist who is Robert Hooke. Robert Hooke, he first discovered the plant cell, the dead plant cell under the microscope. But here it is Robert Brown. He is the scientist who first described the nucleus in eukaryotic cells. Okay. A nucleus, a typical nucleus is covered by a protective layer which is called the nuclear envelope, which is called nuclear envelope, also called nuclear membrane. Okay, so a nuclear membrane, it surrounds the entire nucleus. Okay, it contains the contents present inside the nucleus. Okay, the nuclear membrane, it is actually made up of two membranes. What are the two membranes? One is the outer membrane and another is inner membrane. Okay, so in between the outer and inner membranes of nuclear envelope, there is some space, there is some empty space. What is this space called? This space is called perinuclear space, and the width of this space is between 10 to 50 nanometers. Note down this point, very important for your NEET exam. Okay, so here and there the nuclear membrane is discontinuous. It is having some small gaps here and there. So these small gaps, they form what is called as nuclear pores. Nuclear pores. What is the use of these nuclear pores? Through these nuclear pores, the molecules which are present inside the nucleus can come out. Similarly, the molecules which are present in the cytoplasm can enter into the nucleus. Which molecules can move inside and out means RNA, proteins. These molecules, they will be usually transported from inside to outside and outside to inside through these small openings called as nuclear pores, and you can find such type of nuclear pores throughout the surface here and there, randomly arranged. Understood the concept, students? That is about nuclear pores. Okay, next very important point. The outer membrane of nuclear envelope, it is continuous with the cell organelle called endoplasmic reticulum. It is continuous with which cell organelle? Endoplasmic reticulum, which we have already discussed in the previous session. Endoplasmic reticulum. Very important point. The outer membrane of nuclear envelope is continuous with endoplasmic reticulum. Understood? Next. Inside the nucleus, you can find liquid, a fluid-like material which is called as nucleoplasm. We can also call this nucleoplasm as matrix. You can call it as nuclear matrix. The liquid or fluid which is present inside the nucleus is called nuclear matrix. It is also called as nucleoplasm. In this nucleoplasm, we have two important things. One is the fiber-like structures formed by DNA, which is called as chromatin, and another cell organelle called as nucleolus, which appear as a dark spherical body in the nucleoplasm. Okay, what are the two structures, children? In the nucleoplasm, we can find the slender thread-like structures which are called chromatin, and also you can find a dark spherical body which is called as nucleolus. Okay, so what is chromatin? What is chromatin? In the resting stage, when cell is not in a dividing state, at that time you can find the DNA, it gets folded up with special kind of proteins called as histone proteins and gives rise to slender thread-like structures called as chromatin. So I am writing here the DNA along with special kind of packing proteins called as histone proteins along with non-histone proteins and also some amount of RNA will get folded up to form slender thread-like structures called chromatin. This chromatin is dispersed throughout this nucleoplasm randomly. Okay, it will be crisscrossing. So that is how chromatin is formed from the DNA. So only DNA will not form chromatin. DNA, it also takes help of histone protein, which is a packing protein, and non-histone proteins and RNA, and it gets packed. It gets packed or folded. It gets packed and it gets folded up to form slender thread-like material called as chromatin, which you can see inside the nucleus. Okay, understood, students? So this is how the chromatin is formed inside the nucleus. Then what is nucleolus? A nucleolus is a non-membrane bound cell organelle. That means it is not covered by any membrane. You can see here I didn't draw any line surrounding the nucleolus. That means there is no membrane. So a nucleolus is a non-membrane bound cell organelle. You can write this in your notes. Okay, it is a non-membrane bound cell organelle present inside the nucleus. One more question can be asked. What is the cell organelle within the cell organelle? Nucleolus. Nucleolus is present in another cell organelle called as nucleus. Similarly, ribosome can be also considered as cell organelle within a cell organelle. Why? Because in the chloroplast and mitochondria there will be another cell organelle called as 70S ribosome. Got the point? So such type of questions can be asked in the NEET exam. Okay. Now, what is the function of nucleolus? This dark non-membrane bound cell organelle, it is involved in synthesis of rRNA. What is rRNA? Ribosomal RNA. It is involved in ribosomal RNA synthesis. Okay. So is that always only one nucleolus will be present in the nucleus? No. There may be more than one nucleolus in a nucleus. It depends how active the cell is in process of protein synthesis. If cell is very active in protein synthesis process, then obviously there will be more number of nucleoli. Nucleoli is the plural of nucleolus, which is singular. Okay. So the number of nucleoli present in the nucleus depends upon the protein synthetic activity. If more is the protein synthetic activity of the cell, there will be more number of nucleoli in the nucleus. Understood? Now let us see the points from NCERT book. Nucleolus is involved in rRNA synthesis. Large and more nucleoli present in cells actively carrying protein synthesis. Not only the number of nuclei will be more, but the size of the nucleoli will be also large in the cells which are actively involved in which process? Protein synthesis process. A very important point for your NEET exam. Okay. Interphase nucleus. Interphase nucleus means what? The nucleus which is present in a cell in a non-dividing state, that is called interphase. Interphase is a non-dividing stage, otherwise called resting phase of the cell. So in the interphase, the nucleus has loose indistinct network of nucleoprotein fibers called chromatin. So they are calling this chromatin as nucleoprotein fiber. Nucleoprotein fiber. Why? Because it is made up of DNA, nucleic acid, and also it is made up of proteins like histone proteins and non-histone proteins. That's why the chromatin can be also described as nucleoprotein fiber. Understood? So interphase nucleus has loose, means loosely arranged, indistinct, means not very clear, network of nucleoprotein fibers which is called chromatin. Okay. Chromatin contains DNA and basic proteins called histones. What are basic proteins? So they have basic nature, that's why they are called as basic proteins. They also carry positive charge. Okay. Histones, non-histone proteins and RNA. Just as we have discussed, the chromatin is made up of DNA, histone proteins, non-histone proteins and RNA. So out of this, which are basic? Histone proteins are basic in nature. This is another important point for your NEET examination. Note down. Okay. Chromatin term was given by scientist W. Fleming. So the term chromatin, it was first given by scientist called W. Fleming. Understood, students? Now let us discuss some more points. Okay. Look here. It is very common that every eukaryotic cell will have only one nucleus. But sometimes what may happen, in a single cell more than one nucleus can be also observed. Okay. So for example, this is a protist. Can you identify what is this protist? Paramecium. Okay, in the paramecium there are two nuclei. One is macronucleus, another is micronucleus. So here we have two nuclei per each cell. Okay, this is one special condition which we can see in paramecium. Okay. Sometimes single cell may have more than two nuclei. Okay, example I will tell you. The fungal hyphae. The fungal hyphae. So this is the cell of fungus called fungal hyphae. In the fungal hyphae, in the cytoplasm, you can find multiple nuclei. Multiple nuclei will be present. This is called multinucleated condition. Multinucleated condition. When a cell is having many nuclei, it can be called as syncytium. Syncytium. Note down this point, students. When a cell is having many nuclei, then such cell can be called as syncytium. I will tell you another condition where a cell does not have any nucleus but still it is in living state. Can you tell? Matured red blood cell. Matured red blood cell. Matured RBC does not have any nucleus. It is enucleated. Enucleated means nucleus is absent. So this is which cell? This is fungal hyphae. Fungal hyphae has multinucleated condition called as syncytium. And what is this cell? This is cell of paramecium. Paramecium. So questions can be asked about the nucleus status in these three different types of cells: paramecium, fungal hyphae and matured RBC. So I will tell you one more example. In plants, there is a cell which is in living state but doesn't have nucleus. There will be cytoplasm. Can you tell what is that cell? Yes, it is sieve cell. Sieve cell. The sieve cells are enucleated living cells. Okay, so these are several points, extra points which may be asked in the NEET examination from this concept called as nucleus. Copy down, students. Students, now we'll discuss what are the functions of nucleus. Functions of the nucleus. First, the first function is replication. Replication. What is replication? Look here. It is a process by which the DNA which is present inside the nucleus, it will produce another copy of DNA, which will produce another copy of DNA. This process we are calling it as replication. It will take place inside the nucleus. Okay. Next, the second function of nucleus is transcription. Transcription. What is transcription, students? Look here. The DNA which is already present in the nucleus, it will synthesize RNA, ribonucleic acid. The process of formation of RNA from DNA is called as transcription. This process also takes place inside the nucleus. So replication and transcription, these are the two main functions of nucleus. Understood? Now we will discuss how the DNA can produce chromatin, how chromatin can form the chromosome. Okay. So copy this. I am going to erase. Okay. Okay, students, this is a non-dividing cell. Non-dividing cell means the cell which is not in state of cell division. A non-dividing cell is in interphase. It is said to be in interphase, in a phase called as interphase of cell cycle. So when cell is in interphase, at that time you can see there is a clear nucleus, visible nucleus inside the eukaryotic cell. And inside the nucleus you can see the DNA is in the form of slender fibers called as chromatin. As you can see here, chromatin will be there. The moment the cell prepares to undergo cell division and it enters into phase called as M phase. The cell enters into M phase, means mitotic phase. It is a dividing phase. So when cell enters into mitotic phase, then you can see that the chromatin material which is present inside the cell, it will undergo folding and packing, and slowly it will take the form of chromosome. So after a while you can see that inside the cell the chromatin will condense to form slender rod-like structures called as chromosomes. So these structures are called as chromosomes. Okay. So the chromatin material, it will condense to form structures called as chromosomes. Chromosomes are rod-like structures which are clearly visible under the microscope. Okay. By looking under the microscope, you can even identify the different types of chromosomes based upon their size. Okay. So now, students, now let me demonstrate how chromatin material can form chromosomes by using this rope. So now I have some rope with me. Imagine that this rope is the network of nucleoprotein fibers called as chromatin. So this is the chromatin, something looking like Chinese noodles, right? So this chromatin, which is in the form of slender fibers, it gets folded and get packed, and ultimately it takes the shape of a chromosome which looks something like this. So let's make a proper chromosome. Okay, so the chromatin, it gets packed, it gets packed. Something like this. I'm going to wind it, and again down. Wind it, and winding up. So gradually I'm getting the proper shape of chromosome, children. So beautiful, right? This is the model of chromosome which you can also prepare at your home. Oh, I got some knot here. Remove the knot. Okay. So like this. Yes. Come on, fold it. Yes. And now here I have a proper chromosome. So this is the shape of a proper chromosome, children. This is how the chromosome appears under the microscope. You can see the chromosomes very clearly under the microscope. They appear like rod-like structures. Okay, so a typical chromosome will have two chromatids. You can see here, this is one chromatid and this is another chromatid. Okay, the two chromatids are attached at a common place called centromere. So the centromere is the place where the two chromatids are attached. Okay, so can you tell how many chromosomes you can find in a human cell? Exactly, 23 pairs of chromosomes. So there will be 23 pairs of chromosomes like this in each human cell. Understood the concept, students? If you really appreciate my efforts, then give a big like to this video at the bottom. Okay, now let's continue. Students, now we will discuss about the structure of chromosome. Look here. This is the diagram of a chromosome. What is the definition of chromosome? According to NCERT textbook, it is a highly condensed form of chromatin which is in rod shape. A chromosome is highly condensed form of what? Chromatin, which is in which shape? Which is in rod shape. Okay, you can see here the chromosome is in the shape of rod. It is formed by condensation of chromatin material. That means coiling and packing of chromatin material. So look here, students, I will make you to understand this concept. This is a cell, a human cell. In the human cell, you can see that the DNA is already packed in the form of chromatin with the help of histone proteins, non-histone proteins and RNA. Okay, so this chromatin further it will undergo coiling in next level and finally it gives rise to a structure called as chromosome. So you can see here, this is the DNA, deoxyribonucleic acid DNA. Okay, the DNA, it gets packed with the help of histone proteins. Imagine this is the histone protein. With the help of histone proteins and non-histonic proteins and RNA, the DNA further gets packed into a structure called as chromatin. Okay, so DNA is actually not visible even under microscope, which is that because it is having such a small size. When DNA gets packed, then chromatin, it becomes slightly visible under the microscope in the form of fiber-like structure. Okay, so this chromatin further it undergoes packing. Just now we have discussed with the help of rope. I have demonstrated that. Further the chromatin undergoes packing and forms the structure of a chromosome. Okay, so NCERT book says that in the human cell, in each human cell there will be 2.2 meters of DNA. In every cell of human body, how much DNA you can find? There will be 2.2 meters of DNA, and that DNA is distributed among 23 pairs of chromosomes. So the 2.2 meters length of DNA, which is a very long DNA molecule. So this long DNA gets packed into 23 pairs of chromosomes, that is 46 chromosomes which are present inside a single human cell. So this is a very important point. Note down this. Okay, now let us see the structure of a typical chromosome in metaphase. So this is called a metaphase chromosome. Okay, so in a metaphase chromosome, you can see there are two chromatids. This is one chromatid, this is another chromatid. The two chromatids are attached to a common region attached at a common region which is called centromere. What it is called, my dear children? It is called centromere. Once again I repeat, the two chromatids, what are these? Two chromatids, they are attached to a common center. This region where they are attached at the common center, this region is called as centromere. Centromere. Okay. So these two chromatids can be also called as sister chromatids. Why they are called sister chromatids? Because both chromatids are attached to a common region called as centromere. That's why they are called as sister chromatids. Understood? Now look here. In the chromosome you can see there is a depression. There is a depression at the center. So this depression we are calling it as primary constriction. We are calling it as primary constriction. Primary constriction. So the depression which we can see at the center of the chromosome is called as primary constriction, and centromere is always present near the primary constriction. Understood, students? Now look here, on either sides of the centromere, there are two disc-shaped structure. This is one disc-shaped structure. This is another disc-shaped structure. So these two disc-shaped structures are called kinetochores. What they are called? Kinetochores. What is the function of this kinetochores? Kinetochores, they provide surface. They provide surface for attachment of spindle fibers during the time of cell division. The spindle fibers, they get connected to these disc-shaped structures from both sides during the time of cell division. So that is the function of kinetochores. Understood, students? Understood? Now let us discuss some more points about the chromosome. We can also find another constriction called secondary constriction in some chromosomes. Okay, I am drawing one more chromosome here. So this is a chromosome. This is the primary constriction. In some chromosomes, you can also find the second constriction towards the end, which is called the secondary constriction, which is called as secondary constriction. So just above the secondary constriction, you can find a fragment of chromosome. This small fragment of chromosome is called as satellite. What it is called, my dear children? It is called as satellite. Those chromosomes which will have a small fragment near the secondary constriction called satellite, those chromosomes are called sat chromosomes, otherwise called as satellite chromosomes. Okay, so now another concept we are going to discuss, that is a telomere. Chromosomes also have structures called as telomeres. What are telomeres? Telomeres are structures made up of DNA sequence and proteins that protect the ends of chromosomes like a cap. That protects the ends of chromosome just like a cap. So in the chromosomes, in all the chromosomes, you can see at the ends there are some special DNA sequences at the ends of the chromosome in the chromatids. These ends we can call them as telomeres. Telomeres. So they will protect the ends of the chromosome just like a cap. Imagine this is a pen and this is the cap. This cap, it will protect the nib of the pen. Okay, just like a cap which protects the pen, there are DNA sequences called telomeres which will protect the ends of the chromosome on both sides. Okay, so what are other functions of telomeres? These telomeres, they prevent the sticking of chromosomes. They will prevent the sticking of chromosomes with each other. Okay, if telomeres are not present, what happens? The two chromosomes will come near at their ends, they will fuse with each other. So this will cause a big problem inside the cell. Understood? Okay, the telomeres have a special kind of enzyme it is called telomerase. Telomerase enzyme is also found in the telomere region. So there is one analogy, students, just like the shoelaces. In the shoelaces there will be some protective material at the ends of the shoelaces, just like this. Imagine this is a shoelace. At the end of shoelace there is a plastic tube like structure. Okay, so just like the ends, at the ends of the shoelaces there is a cap like a tube like structure which protects the ends of shoelace. Just like that there are telomeres which will protect the ends of the chromosome. Understood now? Understood? Yes. Now copy down this, students. Students, now we will discuss types of chromosomes based on position of centromeres. So based on the position of centromeres in the chromosome, there are 4 types of chromosomes which you can see here. The first one is metacentric chromosome, submetacentric chromosome, acrocentric chromosome, telocentric chromosome. Now let's see the differences. A metacentric chromosome is a chromosome which is having centromere exactly at the middle. So because of the position of centromere exactly in the middle, there will be 2 equal arms on either sides. So this is one arm, this is another arm. Both the arms have equal length. So then the chromosome is called metacentric chromosome. Understood? Now second type of chromosome, submetacentric chromosome. So what is submetacentric chromosome? Can you see any difference between these two? Look here. The position of centromere is away from the center. The position of centromere is away from the middle. It is not exactly at the center. It is away from the middle region. Okay. So because of this position, there is formation of one short arm called as P arm and one long arm called as Q arm. Understood? Then the chromosome is called submetacentric chromosome. Next is acrocentric chromosome. In an acrocentric chromosome, the centromere is placed towards the end of the chromosome, almost near the end of the chromosome. Because of this position of centromere, there is formation of extremely very small P arm and very long Q arm. Okay, so in this particular acrocentric chromosome, you can see the longest Q arm and shortest P arm. Okay, next is the telocentric chromosome. Here the centromere is placed at the tip of the chromosome. The centromere is placed at the tip of the chromosome. So because of which you can see only one arm. Understood? So these are the 4 types of chromosome. Once again I repeat, in metacentric chromosome the centromere is placed exactly in the middle. There will be 2 equal arms on either sides. In submetacentric chromosome, the centromere is away from the middle region, because of which one short P arm and a long Q arm are formed. In acrocentric chromosome, the centromere is present almost near the tip of the chromosome, because of which there is a very short P arm and very long arm called as Q arm. And telocentric chromosomes, a centromere is present at the tip of the chromosome. So these are the 4 types of chromosomes which you can find in the cells. Okay, copy down this, students. Students, we are going to discuss about the last cell organelle, that is microbodies. What are microbodies? Microbodies are membrane bound cell organelles which are present inside the eukaryotic cells. They contain some varieties of enzymes. You can see here there will be different varieties of enzymes inside these vesicles, and they are present in both plant cells as well as animal cells. Okay, look here the definition. They are the membrane bound minute vesicles. Minute vesicles means small sac like structures which are covered by a membrane. They are covered by a single membrane. Okay, they contain what? They contain different types of enzymes, various enzymes, and where these microbodies are found, they are present both in the plant cells as well as in animal cells. They were first observed by a scientist called Rodin in mouse kidney tubule cells. In mouse kidney tubule cells. Students, there are mainly two types of microbodies, peroxisomes and glyoxysomes. Peroxisomes, glyoxysomes. Now let us see the function of peroxisomes. So these peroxisomes are small minute vesicles which are present inside the cells. What is their function? They will convert harmful poisonous hydrogen peroxide into water and oxygen, which is completely safe for the cell. So peroxisomes plays a very important role protecting the cell from hydrogen peroxide, which is poisonous chemical. And second function is peroxisomes are also involved in a process called photorespiration in plants. So when temperature increases, what happens, students? The plants will automatically start a process called photorespiration, during which they will release a lot of carbon dioxide and there will be no formation of food material. We call that process as photorespiration, and peroxisomes are also involved in this process called as photorespiration. Next, glyoxysomes. Glyoxysomes are also microbodies present in the cells. They will convert the fat present in the cell into carbohydrate by a process called glyoxylate cycle. Okay, so this is about the information about microbodies. We have completed the chapter, students. Play the music. Let us celebrate. Students, in the next class we will meet and we will start one more chapter, new chapter, and I wish all the very best. We will meet in the next class. Until then, be happy. Bye bye.